Welcome back to part 5 of transcription. Uh, Today we are going to be going through the mechanics of all the things we introduced you to last time. So we gave you a list last time of all the different RNA modifications. Now we will be going through and going into detail. So by the end of this lesson, you should be able to describe the processing of ribosomal and transfer RNAs. Compare and contrast the different mechanisms of splicing. Explain how eukaryotic messenger RNAs are modified to have a cap and a tail, and what the purpose of those are. And also describe the process of RNA editing. So again, we have our full list here, and we'll just start diving in and take these on one at a time. So processing is just simply a matter of taking a larger piece of RNA and cutting it into a smaller functional unit. Um, Many of the non-structural genes are initially transcribed this way. So what I mean by non-structural is things that don't encode for proteins. So not messenger RNAs. So tRNAs, rRNAs, and many of the others on that list will get processed in this way. So here are several different chunks that will end up in a ribosome. Um, so if you dig in deep into ribosomal subunits, you'll find out that there is an 18S segment, there is a 5.8 S segment, 28S segment. All three of these chunks of RNA that are going to be a part of a functional ribosome come from one transcript. So you get one overall large transcript, and here the pink regions are going to be cut out and degraded, and the red regions are going to be left as the functional units. Transfer RNAs also have a similar type of processing, and as you recall, a lot of these other types of RNAs have a three-dimensional shape that helps produce a particular type of function. So here you might recognize the overall shape of a transfer RNA. Um, Down here at this bottom loop, we have where the anticodon is. And it is produced from a much larger transcript. Now this is kind of a two-dimensional image of what will become a three-dimensional molecule. This just shows you where stem loops are, where base pairs occur, and so forth. But you can see here that there are larger pieces to this RNA, and it is trimmed down. So one cut is made here by RNase P. This is an endonuclease, so it cuts internal and produces a new 5' end. Another endonuclease actually makes a cut over here. It recognizes the stem loop. And then another DNase D, this one is called an exonuclease. What it does is it starts at one end and starts chewing off. So endonuclease can cut nucleic acids in the middle and just break it up into two sections. Exonucleases start from one end and chew off one nucleotide at a time. So this removes about 9 nucleotides, and then we will be left with this dark red region. Now, what you'll notice is along here you will see other things present. So we have over here that this is not just G, but a methylated G. Over here, you will see something called P, and you might think back, I don't remember P being a nucleotide. Well, that's because we haven't told you about it yet. It is a non-standard nucleotide that is added in or modified later on. So this happens after the transcript is made. Something else would have been in there initially. And here we see a couple other instances of that, and we'll get to that towards the end of this lecture. All right, so that is how transfer RNAs and ribosomal RNAs are processed from a larger transcript and then cut and trimmed. And you'll see here that both of these may undergo further processing than just the clipping aspect of it, or the processing aspect of it. Now, splicing. Splicing has three distinct mechanisms. So there are group 1 intron splicing, group 2 intron splicing, and spliceosome. One distinction here is the spliceosome involves protein machinery. Group 1 and 2 intron splicing are essentially remnants of when splicing was purely an RNA aspect. So we don't need proteins in order to have this type of splicing occurring. We do need proteins for this. By far, the spliceosome is the most predominantly used form of splicing, but we do have remnants of group 1 and group 2 within our genome. In all three of these cases, what the goal of it is to remove intron RNA and then to link the exons back together with a phosphodiester bond. So group 1 and 2 are also referred to as self-splicing because, as I said, they do not require an enzyme. And what I mean by that is it does not require a protein enzyme. It just so happens that the RNA itself acts as its own enzyme. Another word for this would be a ribozyme. Both group 1 and 2 differ in the way that the intron is actually removed and how the exons are reconnected, but they both can occur without additional proteins. You can do this in a test tube. Now, although they don't need additional proteins, there are definitely proteins present in the cell, we call these maturases, that can enhance the rate of this reaction occurring. So you could almost think of it as a catalyst for the catalyst. So let's take a look at these two reactions. What we have here is an exon 1 will have a large intron region and then exon 2. At the beginning of exon 2, we will have a G. In the intron, it folds into a shape that has an internal binding site for a free floating G base nucleotide. This positions the hydroxyl group in such a way to facilitate a reaction to occur. And what this does is it will essentially replace the bond at the beginning of the intron with that G. So notice that there's a G here. That G is now put onto that 5' end, and we now have one end of it free. Now, I don't want you to think that this is actually floating free. It is actually self-bound within this mechanism. So inclusive of having a binding site for this nucleotide, it also has binding sites for this G over here and also the exon 1 site down here. Now, once that takes place, we take the OH group from here. It is positioned next to this G, and you get the similar type of reaction here. This bond will replace the connection point on the backbone with this G, and now this intron is free, and we have a processed messenger RNA. Over here we have group 2. Group 2 is all facilitated through an internal adenine, and that internal adenine essentially carries out the same function as the guanosine does over here, that is a free bound nucleotide. Now we're using an internal one to go ahead and uh, replace the bond, and now you'll see you have a looped piece of DNA. This is still retained within that system and is positioned to replace the bond over here. And now we get a complete messenger RNA, and the intron is then removed and degraded. So here you can see we create a loop. Here we don't have a loop, but we bind a free nucleotide. So both of these mechanisms take place. They are rarer than we see of this next one. So the next form is that which uses the spliceosome. And this is responsible for most messenger RNA splicing. Now, the spliceosome itself has protein components, but it also has RNA components. Real quickly here, we'll just take a look at it. It looks somewhat reminiscent of the type 2 mechanism, in that we have an internal A that forms that kind of lasso type shape, and then it's trimmed off, but this all occurs in the context of a protein backstructure. The spliceosome itself is a multi-component structure, has several proteins, and it also has several small RNAs present. So there is small nuclear RNAs and a set of proteins that interact with those. So these RNAs themselves are produced at a separate location, meaning that they are transcribed from a different gene. Now, these subunits of the spliceosome carry out several functions. They bind to the intron sequence and recognize where the intron and extron boundaries are, hold the pre-messenger RNA in the proper configuration, and carry out the catalysis. What I want you to think about in terms of this is what has essentially been done here is the type 2 splicing mechanism has been turned into an independent operating configuration, and some of the RNA sequences that would have been present in this type 2 alone mechanism are now transferred to this small nuclear RNA subset and carried out in mass that way. So you can think of the spliceosome as the evolutionarily descendant of type 2 splicing. So here we'll dig into the mechanics of this. Here we have an exon and an intron and an exon, and highlighted here are sequences that are of particular importance. So those highlighted in bold are highly conserved, so you see these at almost every single intron. So the GU here, this A is always highly conserved because it is the catalytic site, and the AG over here. Over here where you see Pu, that means that there is always a purine there, but it doesn't matter which one. And over here, um, there's a pyrimidine here, but it can be either one. And here it means that there's a stretch of 12 of those, but it doesn't matter which they are. Um, so these is the actual sites. Mechanistically, what we have, and we've distilled this down to just a few key components. We have this U1 SNRP that binds to this um, junction between the exon and the intron. So it's recognizing the sequence. And again, this is a cluster of small nuclear RNAs and their associated proteins. So this whole SNRP complex we'll call U1. We have a second one, which is called U2, that recognizes the catalytic A. Once those two are bound, U4 and U6 bind in and form a complex with U1 and U2. They also recruit U5, which is recognizing this boundary on the other end, at the 3' end of the intron. Next, we carry out that first enzymatic action. So we get the A attacking the 5' end and replacing it, leaving this as a bound 3' end to this U6 protein. And then further from that point, we carry out the second catalytic mechanism, which will bind this to over here. Now, up here, after that first reaction takes place, U1 and U4 fall off. And then once this reaction carries out, the rest of the proteins are associated with this intron loop, and then the intron will get degraded. So this is the overall process of how most splicing actually works in eukaryotic cells. So it utilizes this spliceosome mechanism. And the spliceosome itself is kind of referred to this complete complex of U1, U2, U4, U6, and U5. All right, another modification that we have is capping. So in eukaryotic cells, most messenger RNAs have a 7-methyl guanosine that is attached at the 5' end. So here we have a messenger RNA that is being produced. This is the 5' end, and we are going to zoom in on it. So here is the first sugar, and the first nucleotide base is here. Here is the 3' end. You've got a phosphate, and then you have a 5' of the next ribose sugar, and it would continue on. At this 5' end, we have a triphosphate because we are adding these nucleotides in as triphosphates. The first thing that happens is we remove one of those phosphates. That is carried out by this enzyme. The next thing that happens is we bring in a G, not a methylated G, just a normal G, and it is brought in as a triphosphate. It is linked on through the phosphates. So we have 2 phosphates here. We're adding in 3 from a nucleotide. And what we end up with is 3 linking the 2 together and 2 having been lost. So 1, 2, 3, 4, 5, and then we'd have 1, 2, and then with the incoming nucleotide, 1, 3, 4, 5. So here we've lost 2. Again, the process of losing these phosphates are energy harvesting steps as well. Now we have a guanosine linked on to the end. And the next step is to methylate it. So we have an enzyme that comes in and adds a methyl to this position of the nitrogenous base. And now we have a 7-methyl. So this is a methyl group in position 7, guanosine cap on the 5' end. Now, this takes place early while the transcript is still being made. So the rest of it is still being produced down at this 3' end. So when it's about 20, 25 nucleic or bases long, it will get this modification put on. This cap is involved in the movement of some RNAs into the cytoplasm and helps act as a marker during translation. So this is one of the things that is recognized during the assembly of ribosomes. And then ultimately it is also a precursor for the splicing step. Things that do not have this modification tend not to get spliced. All right, so this is at the 5' end. What happens at the 3' end? Well, at the 3' end, we get this event called polyadenylation. And what this essentially is a string of adenosines are added on to the end of the transcript. So if you recall from our discussions of how transcription is terminated, we alluded to the fact that there is a polyadenylation signal sequence. So this is a sequence that is in the DNA, it is then transcribed into the RNA, and once you pass this point, you start to see the termination process starting to kick in. Now, this acts as a recognition sequence, and an endonuclease will come in and it will cleave off about 20 nucleotides downstream of that sequence. And then another enzyme will come in and it will only add A's onto the end. So it's not using a template for this, it is just simply adding the A's onto the end. And this is something that can vary in length. The maximum length is about 250 A's added on. And ultimately, the length of the poly-A tail can have an effect on the stability of the messenger RNA. So the longer it is, the longer the messenger RNA lasts before it's degraded. Consequently, you will be able to make more protein from this single message if it lasts longer. So this is yet another way of modifying how much of a protein is transcribed. All right, finally, we have RNA editing. This is the last thing we will discuss in terms of modifications. And this is where you use enzymes to essentially change the RNA sequence. So you come in and you might recognize a particular sequence and mark it for a particular change. This happens in a slew of different species, including mammals. And I'm just going to highlight one change here. I'm going to kind of highlight this A to I conversion. So here we have adenine. We get an enzyme that essentially comes in and cleaves off this amino group and converts it to a carbonyl. And this nitrogenous base here is called inosine. Again, this is not one that you can add in directly, but you can take A's and modify it to this. This is something that you see in several locations. One key place that you see it is in transfer RNAs. And that is because inosine can hydrogen bond to more than one target. And we'll come back and discuss that during translation. So I hope you have a better feeling for how all the different modifications occur. Um, Things that I want you to remember, particularly, are things like the spliceosome. I would have a clear picture of how that works, understand the 5' capping, the polyadenylation. This we'll come back to and discuss more again, so we probably won't talk about this until we talk about the actual function of that. Um, but then also how ribosomal and transfer RNAs are processed or clipped into smaller chunks.